talking about our independent project. We are doing our independent project on the effectiveness of different chewing gum ingredients on oral health. Our objective is to test the effectiveness of different types of chewing gum on promoting oral health. Our three main aims are, first, to discover the effect of high sugar concentration in gum on oral bacterial growth. Our second aim is to discover the effect of cinnamon on oral bacterial growth. And our third aim is to discover the effect of different ingredients, xylitol, sorbitol, or xylitol and sorbitol on oral bacterial growth.
Hi, I'm Sydney, and I'm going to be talking about the experimental design. So we're testing 4 different types of chewing gum, and we're doing one control, which is just going to be gum base. The ingredients have at least one difference in each different type of gum. So our first gum is Juicy Fruit, which has a very high sugar content. Our second gum that we're going to be using is Dentine Ice, and we're going to be using the peppermint flavor, which contains sorbitol, mannitol, and mannitol. Our third one that we're going to be using is Mentos gum, and we're going to be using the fresh mint flavor, which has a lot of S-xylitol in it, and sorbitol, and mannitol. The fourth gum that we're going to be using in our experiment is Trident Cinnamon, which contains sorbitol, xylitol, and mannitol. The fifth gum that we're going to be chewing is just a gum base, and it has no active ingredients, and it's going to be one of our controls.
Hi, I'm Juliet, and I'm going to be talking about the procedure for our independent project. So starting off, we are going to be conducting experiments on every Tuesday. So each Tuesday will be a specific gum, whether it be gum base, Juicy Fruit, Mentos, Dentine Ice, or the Trident Cinnamon. So for our pre-chewing experiments, we are all going to come in and we are going to gargle Listerine mouthwash to make sure that we're all at the same, as a control, to make sure that we're all at the same level, I guess, if someone brushed their teeth earlier or not. So after doing that, each member will swab their own cheek, and then we will streak a TSA agar plate, and then we will perform a saliva enumeration just like we did in class. And then the TSA plates will be incubated for 48 hours, and then the Petri films will be stored in the 4°C refrigerator. After this, each group member will then chew the selected gum for at least 30 minutes. While chewing the gum, it's important not to drink any water or eat any other food to make sure that our results are the same. Then after chewing the gum, we will perform the cheek swab once again, and then streak the plates, and then we will do the saliva enumeration again. So by doing this, we're going to have pre-gum saliva and then post-gum saliva to make so we can compare the microbes in both. So again, the PBS in the enumeration will act as a control for the saliva enumeration. The mouthwash will act as an internal control to make sure that we all are at the same level coming in, and then the gum base will act as a control for the gum. And that's it.
Our fifth,
And I'm going to be talking about the procedure for our gum independent project.
Our second gum is Dentine Ice Peppermint, which has sorbitol, maltodextrin,